Good morning, everyone. This is Pu-Ting, currently an NIH K9 postdoc fellow at AD Forsyth Institute. It is my great honor to be here with you to share with you my current research and also the future research directions I'm going to pursue. The title of my talk today is Probing Life with Photons: How Optical Imaging Uncovers Cellular Machineries of Biological Systems. From the right side of the slide, you can see 4 beautiful snapshots enabled by either fluorescence imaging or label-free chemical imaging. It can be single bacterium, single red blood cell, single fungus, to a group of microbes, such as this beautiful human tongue dorsum microbial consortia that was visualized by multiplex fish imaging. Each color represents a certain bacterial genus. I got my PhD in Dr. Jixin Cheng's lab at Boston University. Therefore, I'm very happy to come back home again. I'm very fortunate to receive multiple awards, as well as publish more than 10 first author high-profile journals over the past few years. If I use one sentence to summarize what I'm doing, that is the synergy between optical imaging and biology and medicine. This synergy can be manifested in 3 aspects. Technology-wise, I have received robust training in developing and the utilization of the label-free chemical imaging modalities, and also the fluorescence-labeled imaging approach to unravel important biological questions. Accordingly, we also developed the advanced image and data analysis approach to interpret the data obtained. With this image as a platform, it enables us to make fundamental discoveries, such as it is unravel this working mechanism of the antimicrobial agent. And in this way, it can allow us to have alternative diagnostic tool or even the development of new therapeutic treatments. For example, diagnosis-wise, we used the transabsorption microscope to visualize and to quantify the glycated hemoglobin at a single red blood cell level. And therapy-wise, we accidentally found that the pigment from one of the superbugs, MRSA, can be activated by blue light photons. Photoactivation of this pigment opens a new avenue for us to kill this superbug with high efficiency and no resistance development. For the future research, I'm going to focus on the functional microbiome imaging in the first few years. Briefly, understand who they are and how they work. I cannot emphasize enough about the importance of microbiome. If we think about the number, there are 100 trillion microbes staying in our body. However, lots of things remain unknown. For example, the relationship between the microbiome dysbiosis and human disease. To understand who they are, we are developing the next generation fish imaging for better microbial visualization, because many microbes remain difficult to hybridize and to be visualized by traditional DNA fish approach. However, with the new approach we are developing, you can see significantly improved visualization efficiency. It is day and night difference. Not only for 2D snapshots, this method works beautifully in the 3-dimensional way as well. Take this 40-micron thickness dendroplex sample as an example. You can see the orientation and distribution of the 2 representative bacteria in the 3-dimensional microbiome bioarchitecture. My second project will be built upon my proposed K99R00 research. We will investigate the microbial adhesive force as a functional attribute. We can envision this as a tug-of-war game, expansion force, and also microbial adhesive force. If the trial force, expansion force, is larger than microbial adhesive force, after expansion, the bacteria will stay away from each other, and vice versa. In this way, we have a tool to differentiate the microbial adhesive force at single cell level. My third project will be building a high-throughput platform for microbiome identity and metabolism analysis. It is the integration of my PhD work and postdoc training. With this label-free chemical imaging for the spatial visualization and the mapping of the chemicals and metabolites to provide us the function information, the multiplex fish imaging will tell us who they are and their identity information. Briefly, we are also rely on the whole genome sequencing or the RNA-seq to tell us their compositional information and also their pathway information. At the same time, we will incorporate machine learning for spectral and image analysis, such as dimension reduction, BM40 denoising, and segmentation, etc. With that being said, thank you for your attention. I'm ready to take your questions. Thank you.